Hi, I am Raghavi from the Department of Biochemistry. Now we see about the topic of coenzyme and its functions. First we know about the what is meant by an enzyme. Enzyme are the large biological molecule responsible for thousands of metabolic process that sustains the life. They are highly selective catalyst greatly accelerating both the rate and specificity of metabolic reaction. Some enzymes require no chemical group for their activity other than their amino acid residue. Other requires an additional chemical component called as cofactor for their required activity. Cofactor. What is meant by a cofactor? A cofactor is a non-protein chemical compound that is required for the protein biological activity. These proteins are commonly enzymes, and a cofactor can be considered a helper molecule that assists the biochemical transformation. Cofactor can be divided into two broad groups: organic cofactors such as flavin or heme, inorganic cofactors such as metal ions and ion sulfur clusters. Inorganic cofactors are some of the examples, that is heme. Organic cofactors. Organic cofactors are small organic molecules. The molecular mass is less than 1000 daltons. They can be either loosely or tightly bounded to the enzyme or directly participate in the reaction. That is NAD, coenzyme A, ascorbic acid, flavin. Coenzymes. These coenzymes are loosely bound cofactors termed as coenzymes. Any of any of the number of freely diffusing organic compounds that functions as a cofactor with the enzyme in promoting a variety of metabolic reaction. Coenzymes are a type of cofactor and they are bounded to enzyme active site to add with their proper functioning. Coenzymes which are directly involved and altered in the course of chemical reaction are considered to be a type of secondary substrate. Coenzymes act as vitamins. Many coenzymes are closely related to vitamins. Some of them are important growth factor. Coenzymes are the precursors of vitamins. A vitamin is a main component of a coenzyme endowed with a biocatalytic function. Coenzymes involved in transfer of hydrogens are called as a hydrogen transferring enzyme, and those which transfers the specific group are known as group transferring enzymes. Coenzymes in the hydrogen transfer reaction, that is a nicotinamide nucleotide biochemical and its biochemical function. First we know about nicotinamide nucleotide. These coenzymes involves in a hydrogen transfer reaction and forms the essential component of dehydrogenase and its function. These NAD plus and NADP are coenzymes of the number of dehydrogenases catalyzing the oxidation reduction reaction. All reactions are catalyzed by them reversible. Next we see about the group transferring coenzyme, that is biotin and its biological function. Biotin is a coenzyme belonging to vitamin B2, a group which is a essential growth factor for yeast and other microorganism but is also required by the higher organism. Biotin is a water soluble vitamin and participate in the transfer of carboxyl group. Role of coenzymes. The function of coenzyme is to transport a group between the enzyme. Chemical group include hydride ions which are carried by coenzymes such as NAD, phosphate group which are carried by coenzymes such as ATP, acyl group which are carried by the coenzymes such as coenzyme A, coenzymes which lose or gain those chemical group in the course of the reaction are often reformed in the same metabolic pathway. For example, in a glycolysis or C3 cycle, NAD plus is involved and it is the main energy currency. Functions of coenzymes. The coenzyme is essential for the biological activity of an enzyme. A coenzyme is a low molecular weight organic substance without which the enzyme cannot exhibit any reaction. One molecule of coenzyme is able to convert a large number of substrate molecule with the help of enzyme. Silent features of coenzymes. Coenzymes are the heat stable. It has low molecular weight substrate. The coenzyme combines loosely with the enzyme molecule, and so the coenzyme can be separated easily by dialysis. When the reaction is completed, the coenzyme is released from the apoenzyme and goes to the same other reaction site. The important coenzymes are alcohol dehydrogenase, coenzyme A, flavin adenine dinucleotide, nicotinamide adenine dinucleotide, and adenosine trinucleotide, FAD, NAD, ATP. First, we know about the ATP. ATP is a function transport the chemical energy within the cell for the metabolism. It is known as an energy currency of a cell. ATP is composed of adenine nucleotide base, a ribose sugar and 3 phosphate group. Energy can be released from ATP when a terminal phosphate group is released in the hydrolysis reaction. This is because the energy of the ATP is held in the bonds between the phosphate group, and when the bonds are broken, it is accomplished by release of energy. Main in the cell metabolism, ATP is used as a main energy currency. Next, we know about the NAD. NAD is composed of 2 nucleotide adenine and nicotinamide. The nucleotide are held together by a pairs of phosphate group which act as a bridge and are also bonded to the ribose sugar each. The function of NAD is to carry the electrons from one enzyme controlling a reaction to another. NAD is involved with the redox reaction because substrate are either oxidized in which they loosely electrons or reduced in which they gains the electrons. NAD is either found as a NAD plus which is an oxidizing agent and is involved with accepting the electrons from the other molecule. NADH which is used as a reducing agent to donate the electrons to the other molecule. Now we see about the FAD. FAD is composed of adenine nucleotide, a ribose sugar and 2 phosphate group. FAD can also exist as monophosphate and is called as flavin adenine monophosphate, FMN. FAD is involved with a redox reaction like NAD. FAD can exist in 2 redox state, that is FAD and FADH. Next, we see about the coenzyme A. Is a prominent coenzyme of a living organism which transfers the acyl group of carboxylic acid. It plays an important role in the metabolism of protein, carbohydrates, fats, which are important reaction that allows the energy from food to released. For example, a coenzyme A is required for the oxidation of pyruvate in the citric acid cycle. Coenzyme A is an important in the synthesis of cholesterol, steroid hormones, and is required for the deoxidation of a range of harmful drugs that can be accumulated in the liver. That is the detoxification. Next, alcohol dehydrogenase, that is ADH, is an energy which is uses NADH as an coenzyme. ADH has a 2 binding region, one with primary substrate ethanol binds and one where the coenzyme NAD plus is able to bind. The enzyme is responsible for conversion of ethanol to ethanol. The reaction is an oxidation reaction, reducing reaction, and result in the removal of 2 hydrogen ions and 2 electrons from the ethanol. The hydrogen ions and the electrons are added to Na plus, which converts the coenzyme to NADH plus H plus. The first reaction involves with the metabolism of ethanol. Thank you.